All right, so as you recall, I told you that just like the basal ganglia, the cerebellum does not directly communicate with either the motor neurons in the spinal cord or with the local circuit neurons in the spinal cord. And the communication is really indirect that way. So as you can see from this picture that is shown here, the cerebellum location is kind of at the back of the brain stem, right below the brain. The word cerebellum really means it's a diminutive version of cerebrum. It means little brain, effectively. If you kind of look at the cerebellum, either from the bottom or from the top, these are the pictures that are shown. The cerebellum is a structure that is developed from the hindbrain. It lies right behind the pons, which is that bump in the brainstem, and is separated from the pons by the fourth ventricle. So remember, these are the ventricles through which the cerebrospinal fluid is flowing. It's connected to the brainstem by three kind of pillars that come in. They're called peduncles, and I'll come back and discuss this. These peduncles, depending on their position, they're called superior, middle, and inferior peduncles. So there's three on each side. And the cerebellum itself, when you look at it, just like the rest of the brain, the cerebrum is two hemispheres, and the two hemispheres are kind of joined in the middle, in this central part, which is often referred to as the vermis. So that's midline is the vermis of the cerebellum. In terms of the anterior posterior structure, the cerebellum has a structure going from front to back. The anterior lobe is kind of in front of this primary fissure, right, that you see here. And then the posterior is behind the secondary fissure, and in between is the middle lobe. So these are really the different lobes of the cerebellum: anterior lobe, the posterior lobe, and this middle lobe, which is sometimes referred to as the flocculonodular lobe. But I'll come back and discuss this when I show you those two little structures that I'm referring to right now. So this is a side cut. This is a sagittal cut here. You can see the cerebellum here. The cerebellum has this very convoluted accordion-like structure, just like the cerebrum, the cortex does. And the one difference is that the bumps that we call in the cortex, we call them a gyrus, each of them. Here they're called folia. So the folia are the bumps, and the sulci, or each of the sulcus that we had in the cortex, they're called fissures. So they're folia separated by fissures. There's not a lot of information here that I'm giving you that is not gross anatomy. Right now, I'm just telling you the gross anatomy of this thing. So bilateral structure from front to back, three different components, and it's kind of very convoluted. And the surface of the cerebellum, this just like the surface of the cerebrum, we call the cerebral cortex. The surface of the cerebellum, we call the cerebellar cortex. And the cerebellar cortex is kind of separated from the middle part of the cortex. I'll come back and discuss that with you. There's another way that one can look at the cerebellum. Instead of looking anterior, posterior, we go lateral to medial. So if you go medial or lateral, the medial part of the cortex is often referred to as the spinocerebellum. The reason for that is that this is the part of the cortex that gets signals from the spinal cord and communicates back to the spinal cord indirectly, not directly. I'll show you how that happens. The lateral parts, which in primates, including humans, are the biggest parts of the cerebellum, are called the cerebrocerebellum. So these are these big lobes on the side. Right, you can kind of see it's big on this side or big on this side. These are the parts of the cerebellum that communicate with the main part of the brain, with the cerebrum. especially with the cerebral cortex, but you'll see they're also communicating with the superior colliculus. Both the cerebral cortex and the superior colliculus, for the discussion of the motor system, have upper motor neurons. In the cerebral cortex, that's in the motor cortex and the premotor cortex, and the superior colliculus has motor neurons, a lot of which communicate for movements of the eyes, and we've discussed that a little bit before. Then, in addition to the spinocerebellum and cerebrocerebellum, there's a part in the back here, which I kind of referred to here as the flocculonodular lobe. This is the flocculus, and this is the nodulus in the center. So the flocculus is the two sides. It's kind of hanging from the bottom, as you can see. The nodulus is in the center. This is called the vestibulocerebellum. As you will see, the vestibulocerebellum is going to be somewhat different from in its type of communication than the cerebrocerebellum and spinocerebellum. As its name suggests, this part of the cerebellum is communicating with the vestibular system or the vestibular nuclei. It gets inputs from the vestibular nuclei that basically tell the cerebellum about the balanced state of the body, and then it sends outputs to the same nuclei to correct the balanced state of the body. So for example, things that are head-eye coordination and movements of the body get corrected by the vestibulocerebellum, and we'll discuss a little bit how that happens. What else do I want to say here to you? So I guess I'll tell you about the peduncles starting here. So if I come here, the connection. So here's the cerebellum sitting kind of on the back side of the brainstem. On the right side, it's shown in clear, so you can see the three columns that go and connect it to the brainstem, which are the superior, middle, and the inferior cerebellar peduncles. In the center of the cerebellum, what you see are, so remember I told you that there's a cerebellar cortex, and maybe I should go back here and point this out. The cerebellar cortex, if you go interior from there, there is white matter, and in the center of that white matter, you have a set of nuclei that are known as the deep cerebellar nuclei. So these nuclei are the main outputs of the cerebellum. So all of the outputs of the cerebellum are going through these nuclei. The rest of the cerebellum, gets inputs and then sends its outputs to the cerebellar nuclei, and those guys communicate out. There's one small exception, and I'll come back and tell you about that. So this is basically showing the structure and the three peduncles. The superior peduncle is almost entirely dedicated to sending outputs from the cerebellum. It sends its outputs primarily from this larger nucleus you see on the lateral side. So the nuclei are on both sides, so this is only on one side. This nucleus, which is pretty big, it's called the dentate nucleus because of it looks like teeth, basically. It's the tooth nucleus. It communicates its outputs to the cerebral cortex indirectly. So it sends its outputs actually to a thalamic nucleus and from there to the cortex. So this peduncle is, again, just outputs, and there's a few of these outputs that go not to the cortex, but go to, as I said, to the superior colliculus. And some of these guys in non-primate mammalian species, the superior peduncle has outputs that go to a nucleus known as the red nucleus, which is also sitting in the brainstem, and I'll show you in a minute what that looks like. The middle part, the middle peduncle, unlike the upper one, is almost all inputs to the cerebellum. And almost all of that are inputs that are coming from the cortex. So the inputs that come from the cortex, maybe I can show you here too. This is the same thing, but shown basically from the opposite side. So here I'm looking from the back towards the front, from the dorsal view. Here I'm looking from exactly the opposite side. So you see the middle peduncle, the inferior peduncle, and the superior one is kind of this small cut that you see here. The middle peduncle is almost all inputs that are coming in, and they're coming from the cerebral cortex, from the neocortex, through a set of nuclei. Again, not directly. They come through a path, and I'll show you what that looks like. So the superior one outputs, the middle one inputs, the lower one, the inferior one, is actually the smallest of these in terms of the number of neurons. Only 5% of the fibers in the peduncles are in the inferior one, but that has both inputs and outputs and communicates mostly to the spinal cord. So it gets inputs from the spinal cord, communicates back to the spinal cord. It also gets the inputs that come from the vestibular nuclei, okay? And also some, since the reticular formation is above the spinal cord, some of the reticular formation inputs, not some, all of them come through the middle peduncles. Those include, for example, the modulatory inputs, for example, serotonergic, dopaminergic, noradrenergic inputs that are coming in, they come through this middle peduncle. So just to summarize some of this, and I have a couple of summary slides that I'm going to show you too. The cerebrocerebellar part of the cortex communicates out through the dentate nucleus to the neocortex, and it receives inputs through the middle peduncle that comes from the indirectly from the cerebral cortex. The spinocerebellar part communicates through a set of nuclei that sit in the middle that are known as the interposed nuclei, and it communicates through the inferior peduncle, and the vestibular cerebellum comes through the fastigial nucleus. It communicates, the inputs come through the inferior peduncle. We'll see that a lot of the outputs to the vestibular cerebellum don't just go through the fastigial nucleus. They also, which is the central ones of these. I don't know if I skipped over that. Let me go back here. I think I had that slide and maybe I skipped over it. Oh, yeah, maybe I did. Okay, so here is the dentate nucleus. I pointed that out. The two in the middle, the two deep cerebellar nuclei in the middle are the interposed nuclei. And the one in the central side is the fastigial nucleus, which is communicating mostly to the vestibular system. So this is, again, repeating exactly what I told you, but showing you the different views of this thing. Now, one of the interesting things about the cerebellum is that each side of the cerebellum, each hemisphere, communicates with one side of the body, just like the cerebrum does. But in contrast to the cerebrum, in contrast to the main part of the brain, this communication with the body is ipsilateral, not contralateral. So the left cerebellum communicates with the left side of the body through the left spinal cord, and the right cerebellum to the right side to the right spinal cord. But when it communicates, when the left side of the cerebellum communicates to the cerebrum, it communicates to the contralateral cerebrum. The reason for that, which should be obvious to you, is that it is the contralateral cerebrum that communicates with the left side of the body. So to be consistent, you have to do your communication that way. So if you think about that, that implies that the inferior peduncle, which communicates with the spinal cord, when it comes out of each hemisphere, it has to go to the same side of the body. It doesn't cross the center. Whereas the axons in the superior and the middle peduncle that communicate out to the cortex or in from the cortex have to cross over. So they do a decussation and communicate with the opposite side of the cortex. Okay? Is this clear? Somebody holler that this is clear what I'm saying. Or if not, ask a question. Is it clear to anyone?
Where does the decussation happen?
The decussation happens as soon as the fibers enter the pons, the brainstem, effectively. So they come in and they cross over and go to the opposite side of the pons to communicate eventually with the cerebral cortex. I haven't shown you how the communication happens, but I just want to make sure that you understand that for the cerebellum to communicate with the cortex, the fibers have to cross. Because cerebellum, each hemisphere is ipsilateral in control of the body, whereas the cerebrum is contralateral. Is that fine?
Yes, thank you.
Okay. Excuse me. So, all right, so let me continue here. So let's talk a little bit about who it is that the cerebellum is actually communicating with. So when you, I'll do this over many slides and I'll come back and summarize, and then I'll pause for you to ask questions about it. Again, only anatomy so far that I'm telling you. The communication, the inputs that come to the cerebellum from the cerebrum, from the, for example, motor cortex. In fact, they come from all parts of the cortex. These parts of the cortex that communicate project their axons to these set of nuclei in the pons that are called pontine nuclei, because they're sitting in the pons. The pontine nuclei are the inputs that go through the middle peduncle to the cerebrocerebellum. Okay, so those are the inputs that are coming in. The outputs that are going from the superior peduncle come from the deep cerebellar nuclei, in particular the dentate nucleus. The ones that go to the cortex, they first go to these two nuclei in the thalamus, which are known as the ventral anterior and the ventral lateral nuclei of the thalamus. Complex is another word for nuclei when it refers to the thalamus. We saw that in the auditory system. So outputs go there, inputs come here. These peduncles that I showed you in the previous slides, so the superior peduncle, just to give you an idea, has about 20 million axons, okay, each of those. The middle peduncle has about the same. The inferior one has fewer. That number, just to put it in perspective, the optic nerve that takes the output of the entire retina, all of the ganglion cells, has only about 1 million fibers. So this is 20 times the number of fibers that are coming out of your eye. Or to give you another example, the pyramidal tract that goes from the upper motor neurons to the lower motor neurons, the main pathway that goes from the cortex to the spinal cord, has only about 500,000 fibers in it. This is one of the biggest bundles of axons anywhere in the brain, with probably the exception of the corpus callosum that connects the two hemispheres of the cerebrum. Okay, so I told you the outputs to the brain go to the thalamus, go to the brain. The inputs come from the cortex to the pontine nuclei and go to this to the cerebellum. There are additional inputs that come to the cerebellum. The inputs that come from the spinal cord either come from these nuclei sitting kind of at the bottom of the brainstem, known as the Clark nuclei. If you remember, when we were discussing I apologize, the somatosensory system, touch and proprioception, that the proprioceptive neurons that are projecting through the spinal cord, so their bodies are in the dorsal root ganglia, they project up the spinal cord, they project to the dorsal Clark nucleus. Okay, that's what that is. There's also touch sensory neurons that project to Clark nuclei, and those are the inputs that come basically ipsilaterally to the hemispheres of the cerebellum. It's kind of interesting because the proprioceptive ones project to the ipsilateral side, which projects to the ipsilateral hemisphere of the cerebellum. The touch and fine sensory, somatosensory inputs, not the proprioceptive, which are the stretch receptors and the Golgi tendon organs, the touch receptors, project to the contralateral. So they decussate, go to the contralateral nuclei in the brainstem, and then those contralateral nuclei, neurons that project to the cerebellum, the spinocerebellar ones, they cross again. So it crosses once and crosses again to go back to the same hemisphere of the cerebellum. So that's called the double decussation. It's a double cross for the touch sensory neurons. The cuneate nucleus takes inputs from the head and neck. So it's similar to what the Clark nuclei are doing, but not for the lower parts of the body, for the head and neck. And the vestibular nuclei we discussed are communicating with the vestibulocerebellum to control the balance. There's another set of inputs that come to the cerebellum that come from this bilateral structure known as the inferior olive or the inferior olivary nucleus. I will come back and discuss that and tell you what it does. So again, we've been going through a lot of gross anatomy, but just let's summarize it one more time. The cortex projects to the pontine nuclei, which project to the cerebellum, okay? There is also an input from the cortex. I haven't told you what this is yet. That goes to the red nucleus, to the inferior olive, and comes to the cerebellum. The spinal cord projects to the cerebellum, and the vestibular nuclei project to the cerebellum. And this is basically showing schematically how that happens. These are the cortical ones coming to the pontine nuclei projecting, the ones from inferior olive, from the dorsal nuclei, and the cuneate nucleus. Now, the spinocerebellum has what's called a fractured topographic or somatotopic map of the body. Mostly, the spinocerebellum is concerned with processing proprioceptive input. So if you think about, think about it in the context of motor activity. If you want to control movement, you better know what the state of the muscles are. And who better reports the state of the muscles than the stretch receptors and the Golgi tendon organs. So all of these guys are projecting and reporting to the cerebellum what is the state of the length of the muscle, each muscle, and what is the state of the tension of each muscle. This projection, there's at least 2 copies of it. There's probably more, but you see one copy which sits on the anterior side of the entire body that is projected there, and another copy that sits on the posterior side, which is projecting the 2 hemispheres of the 2 sides of the body to the 2 hemispheres of the cerebellum. So if you think about it, this part is mainly concerned with the vermis that's sitting in the mid center. These guys are sitting kind of laterally, right? So they're kind of sitting on kind of just lateral to the vermis. You can think about what happens, for example, if there is damage to this part of the cerebellum. So let's say if there's damage to this part of the cerebellum, which has a map of the body that is coming from, let's say, the right hand or the right fingers, it turns out that each part of the cerebellum that's getting input from any part of the body eventually sends its signals to control the outputs to that part of the body, not directly, but indirectly. If you have damage to this portion of your cerebellum, you will have problems moving your right fingers properly. You can't move them smoothly. You will have tremor in those fingers, or you will have rigidness of the fingers. And I'll come back and tell you a little bit about rigidness later on. The job of the cerebellum is to make sure, or as far as we understand it, is to make sure that everything that we do is done smoothly and efficiently, and we become better and better and better at it. All right. If we practice something and we become better at it, be it typing, be it even talking, you know, as you're a baby, as you learn a new language, as you practice a new word, and you say it, and you become better and better at it with a few repetitions, even not over years, just by a few repetitions, that is all done by the cerebellum. This is the spinal part. So you can see the vermis is controlling more of the trunk and the medial part of the body because of the way it's mapped. The lateral side are controlling, the lateral sides of the spinocerebellum are controlling the extremities, including the limbs and also the extremities of the face. Okay. So in terms of the outputs of the cerebellum, the cerebrocerebellum projects to the dentate nucleus. So that part of the cerebellum, the cerebellar cortex, projects to the dentate nucleus, which is the output, which then projects out and eventually ends at the motor cortex, right, which does motor planning. And the spinocerebellar parts project through the interposed and the fastigial nuclei to both the motor cortex and the brainstem. And from those, they project back to the spinal cord to control the movements of the parts of the spinal cord that, you know, is directly projecting back to them, or indirectly projecting back to them. The vestibular cerebellum has somewhat of an exception to this. Remember, this is the part of the cerebellum that was in that flocculus and nodulus. They have axons that go directly out from the cerebellar cortex to the vestibular nuclei. They don't have to pass to the deep cerebellar nuclei. So to the cortical systems, the cerebellar cortex, as I said, projects to the dentate interposed nucleus. It has to cross the midline. These guys decussate, and they go to the thalamus or to the superior colliculus. And if they go to the thalamus, from there to the motor cortex or premotor cortex. So that's basically what is shown here. Okay. There's also an axon collateral to the red nucleus that reports back to the inferior olive. We'll see in a minute what that does. And in contrast, the parts that project to the spinal cord, that go to the brain stem from the cerebral cortex, go through the fastigial nuclei, and they project to the ipsilateral reticular formation, vestibular nuclei, or superior colliculus, and communicate with the lower motor neurons in the spinal cord. My apologies. Okay, so I have a summary here. This is the cerebellum. I told you it's developed from the hindbrain. It lies behind the pons and separated by the fourth ventricle, connected by the three peduncles, and has two hemispheres that are joined in the midline by the vermis, and it has the convoluted folia separated by fissures, and the anatomical subdivisions in one direction are anterior, posterior, and flocculonodular lobes. And this is basically trying to describe the things that I told you in a simple table. I put these as summaries, so you have them in the slides, especially since I'm not in class today. So there's one thing here that is interesting. I just want you to see that from an evolutionary viewpoint, the vestibular part of the cerebellum is the one that evolved first. Okay, so that is part of the, that's why it's called archicerebellum. Archi means historical. So that's the one that evolved first. Then the paleocerebellum or the spinocerebellum, which are the vermis and the regions around the vermis in the spinocerebellum or paravermis. And by the way, globus and umbelliform are the same as I showed you here. I apologize. I'm going to go back for a second here. Yeah, so the interposed nuclei, the one that is round is the globus. The one that is somewhat longer is the vermis. So globus and is the emboliform. Globus and emboliform nuclei are the same as the interposed nuclei, which are sitting in the center. So if you can forget everything and just look at the summary, that should basically summarize this. I also want to mention at this point that all mammals have a cerebellum. All vertebrates have a cerebellum, but the size could be different. The size of the cerebellum could be different in different vertebrate animals. So for example, lizards have relatively small cerebellar structures, whereas for example, fish often have very big cerebellar structures. And in mammals, primates, including humans, actually apes, great apes like humans, have the largest cerebellum. And one of the interesting things is, I haven't told you this, but the number of neurons in the cerebellum is much higher than the number of neurons in the cerebrum, in the main part of the brain. I've told you this in passing, but it's about a 4:1 or 3.6:1 ratio. And that ratio is well conserved across different vertebrates. So there's more neurons in the cerebellum than in the rest of the brain. And I'll come back and tell you a little bit about the neurons. Another thing I wanted to mention is that even in invertebrate species, at least in cephalopods, including the octopus, we know that there is a structure that, in terms of its anatomy and properties, is very similar to the cerebellum. It's the cerebellum equivalent of that structure in cephalopods. All right. I'm going to tell you a little bit about the microanatomy. So what are the cells that are sitting inside the cerebellum now, before I go and tell you about what is thought the function of these things are, the function of the cerebellum is. So, but before I do that, I want to come back and see if there are any questions. I mean, I've thrown a lot of information at you, but it's mostly anatomy information, what sits where and communicates with which part how. And if you look at the tables, you should be able to figure it out relatively easily. So I'm going to continue. If anybody has a question, just holler, just ask. The main cells of the cerebellum that were first studied were these cerebellar Purkinje cells. So here's the diaphyll of one of these guys that you can see. This is the cell body. They're relatively large cells. The axon comes in one direction. But the fascinating part of this, which you can't really appreciate in this figure, but I'll show you in the next slide, is the amazing dendritic structure that these guys have. These are spiny dendrites. So they have little spines, and that means that they get glutamatergic synapses. And if you remember, spines indicate that that particular synaptic input is subject potentially to long-term potentiation or long-term depression. If you think about the dendritic structure, one of the interesting things about the dendritic structure of the Purkinje cells is that it's almost entirely in a single plane. It's two-dimensional. If you rotate it, it becomes like a line. And the cell bodies of these Purkinje cells sit in a particularly well-defined layer of the cerebellum. I'll come back to that in a little bit. Let me show you another slide. So this is a much nicer version of an image of a Purkinje cell. You can kind of see the spines now there and the very, very detailed anatomical structure. On the right, you have a hand drawing of one of these cells by Santiago Ramón y Cajal, who was the known as the father of neuroanatomy, and these were among his favorite cells when he was doing the microanatomy of the brain, which to me is pretty amazing. All right, so in terms of structure, let's go back. The cells, the Purkinje cells sit in one of three layers of the cerebellar cortex known as the Purkinje cell layer. So the three layers, if I start from the outside and go in, are the molecular layer, which I'll tell you in a second, the Purkinje cell layer, which is the cell bodies of the Purkinje cells, and right interior from that is this layer known as the granule cell layer, where these cells, not those cells, these little cells here, the granule cells sit. The granule cells are, unlike Purkinje cells, very small. They only have about a diameter of 5 micron of their cell body, but they are the most plentiful cells in the brain. In a human brain, it is estimated that there are between 50 and 75 billion granule cells. That's between half or to 3/4 of all the neurons in your brain are these granule cells. The granule cells are the main inputs to the Purkinje cells. They actually send their axon up to this molecular layer, and the axon comes up and makes a T branch. So each granule cell goes up, makes a T branch, sends its axon in each direction, each of those branches out about 3 millimeters, total of 6 millimeters, which is about 1/10 of the entire cerebellar cortex. Along this branch, this Purkinje cell can communicate to hundreds, about, anywhere between, I think, 300 and 1,000. The granule cell communicates to anywhere between 300 and 1,000 Purkinje cells, and it makes synapses and goes on. These are not very strong synapses, but they're excitatory synapses, and they're made on the spines. Each Purkinje cell receives input, synaptic input, from about 200,000 granule cells. There's insane amounts of convergence of granule cell inputs to Purkinje cells. Okay, so the molecular layer is where the synapses are happening to the Purkinje cells. That's where the dendrites of the Purkinje cells are. In addition to these cell types, there is a group of interneurons, including the most famous of which is this Golgi cell, which gets its inputs from the granule cells and inhibits the granule cells in turn. So that's a feedback inhibition. It's excited by the granule cell and it inhibits the granule cell. There is also a set of neurons known as stellate cells and another set known as basket cells, and I'm going to tell you a little bit more about each of these. And I also want you to note this thing that is denoted as the mossy fiber here, which is an axon that is coming up and communicating with granule cell. The granule cell itself, that tiny cell there, has about anywhere between 4 to 7 dendrites only. And it gets input from 4 to 7 mossy fibers, but then goes up and communicates to 200,000 Purkinje cells. There's another picture of this shown here. So what you can kind of see here is that the mossy fiber is coming in, communicating with the granule cell. The granule cell sends its axon up, makes a T junction, goes perpendicular to the dendritic tree of the Purkinje cells. So the Purkinje cells are sitting side to side to side to side, and the parallel fibers cut through them and synapse to many of these as they go. The inputs that come from, there's also inputs that come to the, give me one, I apologize, let this call go away. Okay. So the parallel fiber, we understand. That's simply the granule cell communicating to the Purkinje cell. The mossy fiber are the inputs that are coming from the pontine nuclei and also from the other nuclei like the Clark nucleus and the vestibular nuclei. Those are the inputs to the cerebellum. They come in and communicate to the granule cells. So the inputs come into the cerebellum through mossy fibers, talk to the granule cells send their axon, make parallel fibers. Those are the axons that communicate to the Purkinje cells. The Purkinje cells also get another input that is known as the climbing fiber. In fact, this is a very precise input. These are also glutamatergic excitatory inputs. They come from the inferior olive. Each Purkinje cell gets exactly one of these climbing fibers. And I will show this to you in a second. So let's see if I can go. All right. I told you all of this. I told you all of that. All right, so of the Purkinje cells, this, in fact, that should not say the great majority. The Purkinje cells actually don't really project out of the cerebellum. They project to the deep cerebellar nuclei, and the deep cerebellar nuclei project out. There's a few Purkinje cells that communicate directly with the vestibular nucleus, but let's ignore that. The outputs are the outputs of the cerebellum are coming from the deep cerebellar nuclei. And those outputs go, the majority go to the cortex via the ventrolateral thalamus, and some go to the vestibular nuclei, and some go to the red nucleus. And these are just the way they are called. But that's just the name. I'm not going to require you to know it. But now let's come to this thing that I was telling you. The mossy fibers come, those are the inputs to the cerebellum. Talk to the granule cells. The granule cells make the parallel fibers, talk to the Purkinje cell. Each Purkinje cell gets a climbing fiber. This climbing fiber comes from a single individual inferior olivary neuron. It's a glutamatergic neuron. It comes in, it wraps around the dendrites of the Purkinje cell and makes up to 1,000 synaptic contacts with the Purkinje cell. This is one cell communicating to one cell, 1,000 synaptic contacts. This is probably the strongest excitatory synapse in your entire brain. When a climbing fiber produces an action potential, the Purkinje cell always produces an action potential in response. It's excitatory and it's strong. Not only does it produce an action potential, it produces a burst of action potentials, which is known as a complex spike. Okay, so the climbing fiber makes the Purkinje cell produce a particular type of output known as a complex spike, which is a burst of action potentials, right? Whereas these parallel fibers make it fire regular spikes, like simple spikes. The basket cell of the different types of interneuron, which are GABAergic, stellate cells, basket cells, and Golgi cells are GABAergic. The basket cells have very strong inhibition onto the Purkinje cell and shape the activity, the simple spike activity, by producing pauses in it. So that's what the basket cells are supposed to do. Okay, I'm gonna go here. So there's two things I need to tell you that relate to these input outputs. The inputs that come, the mossy fibers that come from, let's say, the pons nuclei, right, from the pontine nuclei. I told you the path that they take to the granule cell, which then parallel fibers to the Purkinje cell, which goes back to the deep cerebellar nuclei. What I didn't tell you is that these same pontine nuclei fibers have an axon collateral that directly excites the deep cerebellar nuclei cells. Similarly, the climbing fibers that go and excite strongly the Purkinje cell, they also have a collateral that excites the deep cerebellar nuclei cell. Ignore what's up here for a second. These inputs are coming to the cerebellum. They excite the deep cerebellar nuclei, which sends an output. The deep cerebellar nuclear cells are also glutamatergic. So glutamatergic excitation out. This is known as the deep excitatory loop. It basically is the shortcut pathway. It's like input comes to the cerebellum says, yes, I heard you, sends an output out. Then this other pathway, this is excitatory. The Purkinje cell is an inhibitory cell. It's a GABAergic cell. So this input comes up here, excitatory, but the Purkinje cell inverts its sign to inhibitory and sends it back to the deep cerebellar nuclear cells. This is known as the cortical inhibitory loop, cortical referring to the cerebellar cortical, not neocortex. And this is thought to be the negative feedback that corrects the mistakes that are being made. What mistakes? Okay, so what is it that this circuitry is supposed to do? Again, I'm throwing at you a lot of information, but let's look at the activity of these cells. Here's a Purkinje cell. At rest, the Purkinje cell fires. So these are action potentials recorded by bringing an electrode nearby the cell body and picking up the spikes extracellularly. These are not intracellular recordings, they're extracellular recordings. The Purkinje cell typically fires with a constant rate of about 50 hertz. That's about 50 spikes per second. Every once in a while, it produces one of these complex spikes, which are marked by these stars. Those have a much lower frequency of about 1 hertz. So you get about one of those per second. So this is a typical firing of the Purkinje cell. And in fact, if you remove all synaptic input from the Purkinje cell, it's going to fire at about 50 hertz like clockwork without any variability. So all of the variability that you see is due to synaptic input that it's receiving. Now, here's a recording of that same cell as this is in a monkey, as the monkey does wrist flexion and extension. So basically is doing, flipping the wrist over back and forth. And you can see as the wrist is moved back and forth, the activity of this cell is completely modulated by the wrist flexion and extension. And in fact, is predictive of what the next move is going to be. So the cerebellum is basically predicting what the next move is going to be of the wrist flexion and extension. Similarly, at the output level, the deep cerebellar nuclei at rest fire tonically at about 30 hertz. But again, excuse me, flexion and extension modulate their activity to go up and down with the motor activity. This is the first indication you've seen that the cerebellum has something to do with motor activity. Now, what exactly does the cerebellum do with the motor activity? To give you an example, a lot of the examples we know come from simpler movements which are easy to record, especially in monkeys when people started looking at this. So this is an eye, and these are the eye muscles that move the eyes left and right and up and down, right? So every time you, let's say, saccade, you move your eye using these muscles. In this experiment, there was a partial lesion, a partial sectioning of this tendon that moves the left eye to one direction, right? So the lateral rectus tendon was partially lesioned. So when the animal is saccading to a particular point, the right eye moves to the position correctly, but the left eye undershoots the target, okay? Now, on the animal, they put a patch on the right eye, and they ask it to saccade, to move its eye to a target. The animal does this and gets usually a drop of juice as a reward when it does this correctly, when the eye moves to the correct position. After about 5 days of doing this, the animal compensates and learns to basically move this eye to the position that the saccade is supposed to go. So let's say a dot appears. So the animal fixates on a point in the screen, a dot appears somewhere else in the screen, and the animal is supposed to saccade to that point, and the head is fixed with a fixture, so the animal can't move its head. And it learns to do that. And if then you move the patch to the left eye, you realize that in the same duration, the right eye is now overshooting where it was supposed to go. So the animal has learned, because the right eye was covered, to compensate for the left eye, but the right eye is overshooting. So in time, the left was corrected and the right is overshooting. This property of learning a new position for the saccade when the muscle is weak is blocked by blocking activity in the cerebellum or the cerebellar cortex or the cerebellar outputs. Okay, if you lesion, let's say pharmacologically, the cerebellar peduncles, the animal will not learn to do this. So this is saccadic eye movements and the process of learning that is done entirely by the cerebellum, or in big part by the cerebellum. Another activity that we know that is kind of in the same vein is the vestibulo-ocular reflex. We talked about this in the last lecture. That's the reflex in which your eyes stay fixed on a location while you move your head up or down or left or right, right? So that's controlled primarily by the superior colliculus, which controls the muscles to the eye and the neck and head. And in an animal that is normal, you can put a pair of glasses known as minifying glasses. So if you know what magnifying glasses are, minifying glasses are the opposite of that. So when the animal is moving their head, the field that they see in their vision is moving less, is moving more than they expect. So the eyes move too far in relation to the image. Okay, so, and basically the vestibulo-ocular reflex that keeps the eyes fixed is known to be related to what's known as the retinal slip. So if you look at the image that the retina is getting from the outside world, if your eyes are fixed on a point in the outside world, as your head moves, the image on the retina doesn't change much. But if your eyes move with your head, the image slips on the retina. That's called the retinal slip. And that's what this superior colliculus compensates for. After several hours of the animal having these glasses on, the animal learns to compensate for the glasses. And then as the head moves, the eyes remain fixed despite the fact that the vision is not matched with the normal vision that the animal got. So it only gets several hours for the animal to match that. And guess what happens now if the animal doesn't compensate? I'm sorry, if the animal's glasses are removed. Well, if the glasses are removed, then the animal compensates in the opposite direction by not moving the eyes enough, because it's learned to move them to overcompensate for the with respect to the normal movements. I think I'm going to stop now because we have 2 more discussions and I'm going to finish this lecture in class. I only have 4 more slides to go, but I'm going to finish this in class and maybe answer some of your questions that came up about this lecture.